Sciences at the University of Queensland. So welcome, Kirsty, and thanks very much for presenting your findings here in this seminar. So I hand over to you, and we are all excited to see your talk.
Thank you very much. So today, I think I'll probably give you a little bit of a different perspective because I'm by training a virologist, immunologist. So certainly I don't have the technical or statistical capacities of many of you, but hopefully this work will still be of interest to you. So what I want to present today is some of the work that we've done really since the start of the COVID-19 pandemic, looking at the role of children in the transmission of SARS-CoV-2. So from the very start of the pandemic, this was an incredibly controversial topic. We've known for quite some time that children are the main drivers of influenza in the community. So this is one of the reasons that children are actually highly recommended to be influenza vaccinated, not just to protect themselves, but also to stop the transmission of the virus in the community. So when SARS-CoV-2 came along, I think there was a temptation to think, well, this is like any other respiratory virus that we see. Children are going to be the main drivers of the virus in the community. And accordingly, there was lots of suggestions that children were maybe asymptomatic spreaders of the virus, that we should immediately close schools, which is a typical pandemic response in the context of influenza. But others were suggesting that, the early evidence wasn't amounting to much in terms of children spreading the virus, and that we need to prioritize keeping schools open. So I think it was a very, very controversial topic, and it's something that we thought we wanted to look into. So this was at the start of the pandemic, and what we did is we collected basically data on household transmission clusters of SARS-CoV-2. And what we wanted to do is use these clusters to determine how often was the child the index case. How often was it the child bringing the virus into the home and then spreading that to the rest of the family? So we went through all the household transmission clusters that we could find of SARS-CoV-2, and we just did this simple table where we looked at the number of cases, household transmission clusters with children as the index case, and the number of household transmission clusters with adults as the index case. And what you can see of the 213 clusters that we found that met our selection criteria, only 8 had children as the index case. And these children typically had less secondary cases compared to the adults. So this was sort of very early evidence that maybe SARS-CoV-2 and influenza were quite different, and that it wasn't children driving the transmission of this virus. But of course, there's lots and lots of caveats to these data. So we need to take those into account. So the first possible criticism of these data is that, okay, well, what we know is that children can be relatively asymptomatic insofar as children typically have a much more mild presentation. So it's possible that when we look for the index case in the cluster, that we're not identifying children as the index case because they're being asymptomatic. So what we did is we went through the household clusters and we identified cases where there was an asymptomatically infected child in the family. And we just erred on the side of caution and said, okay, well, let's assume worst case scenario that that child is in fact the index case of the cluster and they've been misidentified. So even if we did that, and even if we took that into account, we still had children being the minority of cases of the index case in the cluster. So we don't think that's confounding the data. Another possible explanation for these data is, well, we know that at the very start of the pandemic, especially in China, where a lot of our data came from, infection was typically associated with travel to one of the endemic areas. So Wuhan and later the whole province of Hubei. So what we thought is, well, maybe these data are just reflecting the fact that an adult is more likely to actually travel on their own, whereas a child isn't. So what we did is, again, we stratified the data and we took out cases where either, we only focused on cases where either the whole family had traveled or the whole family was in an endemic area to control for that confounder. And again, even if we did this, what we saw was that children were still the minority of index cases in the cluster. Now, the other possible suggestion, as we know, early on in the pandemic, there was a lot of school closures. So there was a possibility that we weren't seeing children as the index case in the cluster because they were simply staying at home. And, you know, unlike adults who may have essential jobs, they weren't mingling with the community. So to control for this, we only looked at clusters in countries where children were still going to school. So children were still out and about. And again, even if we do that, what we can see is that children are still not frequently reported as the index case of the cluster. Just to give you a point of comparison, and if you remember, I told you that children are great at spreading influenza. To sort of validate this method that we were doing, we did the same thing, but we looked at cases of avian influenza because they're frequently reported and well detected. And when we look at influenza, what we see is that children are actually the index case of the majority of household transmission clusters. So there really does seem to be this difference between the role of children in the spread of influenza and SARS-CoV-2. So the next thing we wanted to do is to use these data to say, okay, if there is an infection within the family, how likely is a child to pick up that infection? How likely is a child to contract that infection? So we did this looking at children versus adults. And in fact, what we found is that children were significantly less likely to contract the infection if it was in the family compared to adults. We didn't see a difference between younger children and older children, but to be honest, we had very low numbers in this cohort. So it's very possible that this is just due to the reduced power of this test. So what these data suggest is that if a child is infected, the infection either doesn't take hold or it's rapidly resolved. As a result, the child is not frequently the index case of a household transmission cluster. If somebody else in the family is infected with SARS-CoV-2 and brings the virus into the family, children are much less likely to acquire it from their household contacts, their household cohabitants. So this really raises the intriguing point of why would children be more resistant to infection? What is it about children and maybe the pediatric immune response that mediates this differential response to SARS-CoV-2? So we started thinking about this, and we were wondering if there was something fundamentally different in the antiviral response of pediatric nasal epithelial cells compared to those of adults. Remembering that nasal epithelial cells are the first point of entry for the virus. So the virus comes in and it infects your nasal epithelium, and this could be an important point of difference. So what we did is we took nasal epithelial cell swabs from adults and children, and we grew them at an air-liquid interface. We then studied them and eventually infected them with SARS-CoV-2. So the first important point to note here is that even in the absence of infection, pediatric and adult nasal epithelial cells are fundamentally different. First of all, you can see this by histology. These are the adult nasal epithelial cells grown on a transwell membrane, and these are the pediatric nasal epithelial cells. You see both have these goblet cells, ciliated cells, they're producing mucus, but you do see that in the pediatric cells, we see these pseudocytes, which is obviously clearly a different morphological indication than what we see in the adult cells. To further explore these differences between pediatric and adult cells, we performed RNA-seq on these cells. And what you can see by the PCA analysis is that based on their gene expression, and again, this is in the absence of a viral infection, the adult cells sort of cluster together and the pediatric cells sort of cluster together. And in fact, we see numerous differentially expressed genes between these two different cell populations, some of which includes aspects of the immune response. When you look at these cells by microscopy, you can also see there are some differences. So this is staining of adult and pediatric cells. You can see here staining for DAPI, which is just showing you the nuclei of the cells. Here you can see zona occludens 1, which is an indication of well-differentiated epithelial cells. So you can see really nice staining here, which tells us that these cells are indeed forming a good epithelial barrier, much like what you see in the nose. You can also stain these cells for MUC5A, which is a mucin, and this indicates that these cells are producing mucus. And that's exactly what you would expect in the nose, because these cells do produce mucus in the nose. Here's some phalloidin staining, which is just telling you the integrity of the cells. And importantly, here's some immunofluorescence staining for ACE2. So ACE2 is one of the receptors for SARS-CoV-2. And there's been some reported differences that the expression level of ACE2 is different between kids and adults, and that maybe this explains the reason that kids are less susceptible to infection. But these data have been really, really controversial, and they've been really, really inconsistent between studies. So we looked at this here, and what you can hopefully see here visually is that we do indeed see more ACE2 in the adult cells compared to the pediatric cells. And you can see this in both our representative samples. So we wanted to explore if this was the reason that maybe kids were more resistant to SARS-CoV-2 infection. So what we did is we essentially repeated these experiments, but we looked at the expression of ACE2 by Western blotting. And here it's a bit difficult to interpret, to be honest with you. We do see a trend towards more ACE2 expression in the adult cells, but certainly this is not significant between groups. So what we would suggest is that maybe there is a slight difference in ACE2 expression between the pediatric and the adult cells, but that this is not consistent between donors, and it's probably not enough on its own to explain the differences in infection that we see. So the next important step was to check that our epithelial cells could indeed mimic the clinical situation. Can we indeed see more viral replication in the cells of the adults compared to the kids? So we infected these cells with SARS-CoV-2, and we looked at viral replication over time. And hopefully what you can see here is that when we look at viral titers from these cells, there is significantly more virus replication in the adult cells compared to the pediatric cells. And you can see this when you look for infectious virus, but you can also see this when you look for viral proteins by Western blot. Remember back to what I told you about the role of influenza and pediatric infections, and that we know that kids play a really important role in spreading influenza. So we wanted to sort of contrast this in our in vitro model. So we did the exact same infection experiments, except this time, instead of using SARS-CoV-2, we used influenza virus. And here you can see a really different pattern. You can see, if anything, the virus replicates more in the pediatric cells compared to the cells of adults. So this is sort of consistent with what we know happens in real life, that kids aren't spreading SARS-CoV-2 as much, but they certainly play an important role in the spread of influenza. So maybe this is explained in part by differential ACE2 expression, but there could be other explanations. And specifically, one of the other explanations we wanted to look at was the host response to viral infection, and specifically the sort of antiviral response. So what we did is we took our pediatric and our adult epithelial cells that have been infected with SARS-CoV-2, and we looked at what genes were being switched on in these cells and what genes were being differentially expressed between our adult cells and our pediatric cells. And in fact, what we saw was really, really striking. When we look at these differentially expressed genes, we see only in the pediatric cells are we getting these genes associated with the antiviral response being switched on. So we're seeing lots of interferon, inflammation genes, all these things that we know are controlling the viral infection. In contrast, in the adult cells, we're not really seeing much of a response to the viral infection. So we started hypothesizing that maybe one of the reasons kids are more resistant to SARS-CoV-2 is just that fundamentally their nasal epithelial cells can mount a better immune response to the virus. And in fact, we confirmed this by qPCR, so just a different technique looking at the same thing. And again, when we look at SARS-CoV-2, we can see that at 48 hours post-infection, we're getting a lot more of these antiviral genes being switched on in the kids compared to the adults. Of interest, if you do these same experiments with influenza, you also see more of an antiviral response in the kid cells compared to those of the adults. So probably there is just something fundamentally different about the immune response of pediatric cells compared to those of adults. And this is sufficient to control SARS-CoV-2 replication in the nose, but it's probably not sufficient to control influenza virus replication. And that's consistent with the literature we know about how important this antiviral response is in controlling these different viral pathogens. So these experiments were all done with the original strain of SARS-CoV-2, but of course, as we know over time, new virus strains new viral variants have emerged. And there was lots and lots of concern since the emergence of the Alpha variant that these were actually suddenly preferentially targeting children. So this is a study on the Alpha variant, which is the variant formerly known as the UK variant. And in this study, they were arguing that all of a sudden they were seeing these large number of pediatric infections. This has similarly been suggested with the Delta variant, and you've probably seen that in some of the media coverage, in particular of the recent Sydney outbreak, where we're seeing suggestions that all of a sudden this virus is preferentially targeting children. So we wanted to test that in our experimental model. Now, we've only just received the Delta variant, so we're just growing up the Delta variant at the moment. So we didn't have that available at the time of these studies, but we did have the Alpha variant or the UK variant available. So what we did is we performed exactly the same experiments, but this time using the UK variant. And what you can see here on the left is once again, we're seeing very strikingly that the virus is replicating better in the adult cells compared to those of the child. And once again, when we look at the interferon response or the antiviral response of these cells, we're seeing a higher antiviral response in the cells of children compared to those of adults. So it seems to be, I mean, we can't verify this yet with the Delta variant, as I mentioned, but this seems to be a generic factor across different SARS-CoV-2 variants, that these epithelial cells from kids are just fundamentally different to the epithelial cells from adults, and that this is mounting a different immune response to the virus. So just to conclude, hopefully what I've convinced you today is that children are really unlikely to drive the household transmission of SARS-CoV-2, and this is because they're less likely to be infected with the virus. And probably one of the reasons that these children or this population group is less likely to be infected with the virus is because of just fundamental differences in the nasal epithelial cells of children and adults. So finally, just to conclude, I'd like to thank everyone who made this work possible. In particular, special thanks has to go to my PhD student, Yanshan, who's been driving all this work, as well as our various collaborators and my group at the University of Queensland, which was previously known as Flu Crew, but then we had to update our name because SARS-CoV-2 came along, so now we're Flu Core. Thank you very much.
A great talk, Kirsty. So we have 10 minutes to ask questions. We kindly ask participants to keep your questions short and concise. So maybe I will start with a question. So what were the age ranges for your pediatric and adult groups? I saw in one of your slides that the kids were younger than 20. So is that the whole the age range that you use for entire experiments?
Yeah, so most of our kids were aged about 5 to 6, so quite young.
Yes.
They were all under 10 years of age. Our adult populations, generally, our adults were aged around 30 or so. We did do a separate step of studies seeing if we saw different responses between adults aged around 30 and adult aged plus 60 plus. But we didn't really see a marked difference in those two populations, aside from the fact that the epithelial cells from the older adults probably grew a little bit less and they were just a bit more fiddly. So you could see that sort of phenotypic difference, but in terms of replication of the virus, older adults and younger adults seem to be largely the same. So it was really these under 10s that were quite different.
All right, thank you.
Can I ask, hey Kirsty, thanks for the nice talk, interesting talk. So I have a few questions. I'll ask one first and then maybe one later. So obviously in terms of policy, as you said, this was, you spoke here about the biology, but can you share some views and thoughts and how this is informing policy? If at all, I understand that some views are subjective.
Yeah, so we were actually very surprised going into this research about how controversial it was. And the role of children in this pandemic has been a very emotive argument, I think, because obviously everybody wants what's best for the children, but I think a lot of parents were really scared about the risk that this virus posed to children. So based on these data, we actually wrote a piece in The Conversation arguing that, especially in Australia, where COVID cases were very low, the priority should be on opening schools, because based on our findings, we would say that the risk of not having schools open, in terms of the risk it poses to child welfare, mental health, development, all those sorts of things, was actually much greater than the risk that they posed in the transmission of the virus. Now, that balance does shift somewhat when we're talking about countries where there was a large community caseload of SARS-CoV-2. But even in those communities, we were really advocating that schools should be the priority thing to open, that if you're talking about what to open first, it should be schools. And in fact, our data was actually used by the UK government to justify their policy of opening schools in the, gosh, I've lost track of the waves of the pandemic, but I think in the second wave of the pandemic. And you'll note that they did indeed open schools, and that didn't exacerbate the spread of the virus. So there is a way to ensure that kids can still get face-to-face learning, even in the context of a viral outbreak. But I would gauge this with caution and say that if this was an influenza pandemic, the story would be very different.
Yeah, thanks.
Can I ask a quick questions? I'm sorry I popped in a little bit late, but it's a very quick question. So what do you think about sort of recent COVID-19 variants such as Delta? How bad it could be on children, and I think that there's evidence that children are more susceptible to variants. And what do you think about it?
Yeah, excellent question. And this is something that, like I said, we're growing up the Delta virus now to answer this experimentally. I am actually very skeptical of the reports of Delta being more infectious to children or worse in children because of two factors, I guess. The first is a lot of that data comes from the UK. And when we're looking at that data, that's from a country where the only population that is not vaccinated is young children. So is it that you're actually getting more infections in young children, or is it just we're seeing more infections in the unvaccinated population who happen to be young children? The second thing I would say, and this is why I'm very cautious of it, there was the same debate around the Alpha variant when it first emerged. And again, there was lots of arguments saying we're seeing more cases in children, but in fact, what the situation there was is that overall you were seeing that the Alpha variant was more transmissible. So you were getting more cases in children than we had previously seen, but you were also getting more cases in adults than we'd previously seen. So it wasn't that the virus was preferentially targeting children, it's just that it was inherently more transmissible for everyone. So there's no actual good experimental data on Delta and the effect that it has on children. I would reserve judgment until I've seen the data. And I still would say, even in the current outbreak, you know, again, school should be the priority. And it may be that we have to put in more mitigation strategies because this is a more infectious or more transmissible variant. So maybe, you know, you need to look at bubbling kids or low class number sizes or things like that. But I would still argue that getting face-to-face learning for kids, especially younger children, has to be a priority. I see there's a question in the chat, so I can answer that if you want me to, Hamid.
Yes, yeah, definitely. I was going to read it for you, but
oh yeah, great. Yeah. Go for it.
But now you can answer that.
Maybe I should ask it verbally because I don't think I wrote it right. So you were saying that influenza and SARS-CoV-2 both present with an elevated immune response in children. Is that right?
Correct.
Okay, but you're saying that influenza is dominantly spread through children, where SARS-CoV-2 is not. I was just wondering, I didn't understand why that's the case.
Yeah, excellent question. What we think, and what's been shown before, is that SARS-CoV-2 is really susceptible to this antiviral response. It's very good at blunting the replication of the virus. Whereas influenza is a different virus and that has evolved different mechanisms, it's much less susceptible to this antiviral response. So if you imagine you have the two viruses, there's an antiviral response, that's enough to stop the replication of SARS-CoV-2, but it's not enough to stop the replication of influenza. So that's why we think that probably there is a differential role of children in the spread of these two viruses. Does that make sense? Yeah.
So you're saying that children are naturally more susceptible in general to these kind of to viruses, except that SARS-CoV-2 is uniquely weak to that immune response?
Exactly.
And is it all possible for variants to occur that feature a similar, I guess, profile as influenza?
Yeah, it is a really interesting question. So this susceptibility to the antiviral immune response, it's only been looked at very, very superficially in the variants that we've seen to date. There is some preprint evidence, it hasn't been published yet, that maybe these variants are indeed evolving away from this susceptibility to the antiviral immune response. But it's still very early days. So I think this is something really important we need to watch. And it's not something that many people look at because most people with the variants are focused on what effect does this have on your antibody neutralization capacity, because that's what we know is important for the vaccines. But you also have this whole other arm of your immune response that needs to be considered. So it is theoretically possible. Anything's possible with viruses. That's kind of what makes them cool, I think. But we haven't seen strong evidence for it yet.
Thank you.
Got a quick question?
Yeah, sure.
Hi, Kirsty. Great talk. Really enjoyed that. My question is actually very general. I just don't know anything about experimental virology. And I'm kind of wanting to know the scope of what you can do in the lab with in vitro samples to investigate other aspects of COVID. So I'm working on a kidney study that's looking at just incidence of acute kidney injury, which is like, you know, heart attack of the kidney in COVID, and the rates are very, very high. So when we want to put a little bit of a story around what can, you know, what's actually going on in the kidneys, you've got the option of doing biopsies in people that are very sick or doing autopsies in people that die. But I don't know what the scope is for experimental virology, for taking in vitro samples. And can you give me a bit of an idea about what that?
Yeah, very easy. So our approach to virology, we're certainly not sort of molecular people. We're not the group that looks into one particular path or anything like that. What we tend to do is look at what's happening in the clinic, try and model it in the lab, and get some information that we can then either feed back into public health or clinical care. So these are the sorts of experiments we do routinely. So if you were interested in looking at the role of SARS-CoV-2 in kidney disease, we could either do that in vivo using an animal model, which, you know, there's pros and cons to that, or we can do it in vitro using human cells. So you could, the most simple experiment would be that you could get kidney cells, expose them to the virus, see how they respond, see how the virus replicates. Or you could do a slightly more complex model, which is certainly what we've done looking at responses to the virus in the heart, where we sort of create a culture of the lung. We infect the lung cells, and then we take what we would consider the serum, or the blood from that, because that's sort of what would be circulating and touching the heart, and then transfer that over to our cardiomyocytes and look at their response. So there's all different levels of complexity that you can do, but it's certainly, this is something that we've been really pushing to try and combine the clinical data with the experimental data, because at the end of the day, you have different pros and cons. The clinical data is great for answering some things, but as you say, often the samples that are available are autopsy samples. So you're selecting on a group of extreme patients. So if it is something you're interested in, I'm definitely happy to chat with you, and we can think up an experimental plan, because this is our sort of bread and butter.
Yeah, that would be wonderful. I mean, what we're seeing is that it's most of the burden is actually very early in the illness, which is, this is the kind of new finding. So it would be really interesting. Yeah, I'll send you an email and we'll have a chat offline. That'd be wonderful. Thanks, Kirsty.
Yeah, fantastic. I love the links between clinical and basic virology. It's good.
Great.
I think Zoltan has a question. I can see his hand. Yep.
I have a short one. So kind of two questions related. So one question is, so you showed that kind of children don't seem to play a very significant role in transmission and like health consequences for COVID. So what would that imply in terms of vaccination? What do you think? Is it essential, or is it useful to vaccinate children eventually? Of course, now is not relevant yet in Australia. The other question, is probably more well-defined question, is we know that there is kind of an exponential age dependence on the mortality of COVID infection. And so you were talking about children as a separate category. And so the question is it possible that this can be extended a bit on a broader age group, that the response on nasal epithelial cells could be different even in up to a certain age, even in young populations? Of course, at older age groups, there are other issues that are unrelated due to other diseases and other things. But is it possible that there are differences in this nasal epithelial cells even at up to a broader age range, and that can affect mortality?
Yeah, two really excellent questions. So I'll start with the first one about vaccination. I find this debate very interesting, because when we talk about vaccination for flu, for me, it's a no-brainer. Kids should be vaccinated because it protects the rest of the community as well as kids. When we talk about SARS-CoV-2, I don't think we'll have, based on the data I presented you, I don't think we'll have such a significant effect on the community transmission. So then the benefits of vaccination has to be, does it protect the children? And I guess what we know is that children are at low risk of severe disease, but they're not at no risk. So I could imagine if it was my child, and they could get a vaccine that protected them against something, even if it was a 1 in 100,000 or 200,000 event, that would be something that I was opting for. The second thing that I would say is that we still have questions about long COVID in children. The data is not really there yet to understand to what extent this persists in children, but it would be certainly something that I would flag, and it's certainly something I consider in my own assessment of the value of vaccination as an individual who doesn't have underlying comorbidities, who's not in the age group where COVID is likely to kill me. My primary concern is actually long COVID. So I think those two things have to be factored in. The other thing that has to be factored in, and we don't have the data on this in terms of younger children, we only have the data for children as young as 12, is what the side effects of these vaccines are. What we're seeing with the Pfizer vaccine, and this is basically only really been shown with the mRNA vaccines, is there is an increased risk of myocarditis in younger individuals. That's potentially a point of concern, but that seems to be very, very mild and transient and very, very easily treated. So in my mind, there is a clear benefit for vaccinating children as low as 12. Until we see the safety data for children that are younger, I think it's difficult to make that equation, but those are the sorts of things I would factor into the decision. In terms of your second question, it gets to the heart of this question of immunologically, when is a child a child? We define children as individuals younger than 18, but it's not like the second you turn 18, your immune system suddenly switches and you have a differential response. So what we've seen in a lot of the epidemiology data and a lot of the data that we've generated is really the most protective effect that you see against SARS-CoV-2 is in children younger than 10. So that's why we sort of erred on these on the younger side of using these cells, as I answered before, because that was where we thought we'd see the best phenotype. I suspect if we went too much older, so if we started going into teenagers, probably the cells would start to resemble the adult phenotype, because I think it's really the under 10s that are the most protected. And again, this weighs into your first question about the vaccine decision-making, where we're now seeing people down to the age of 12 being protected. And I think that's very logical. Whether under 12 needs the same level of protection, I don't know. But yeah, I would suspect that immunologically, sort of 10 is your definition of a child.
So, for example, if we would compare young adults to like 20-year-olds to 30-year-olds, so do you think that these gene expressions would already be similar, would not have anything left from the children, so this like switches after a certain age and then it settles to an adult form?
Yeah, so I think our youngest adult was maybe 20 in the low 20s, and we didn't really see anything different in that adult compared to some of the donors that we had that were over 60, or even donors that we had that were in their 30s. So I do think once you reach an adult phenotype, you're probably stuck at an adult phenotype, and unfortunately, yeah, immunologically, you're an adult, like I said, above 12. So yeah, none of us are getting any younger, unfortunately.
Okay, thank you.
Hey, Kirsty, I wanted to squeeze in one more question, and that's with a kind of mutation and in what way you think that virology lab data can inform softer AI, general data science models, etc. So here's the basis for the question. I think it's quite obvious that if the more interface there is with children, the more that mutations can latch on and adapt themselves to strands that will actually persist in children and potentially be harmful. I assume that's kind of given, right? I mean, that's that hypothesis. And
it's
the
theoretically possible, yeah.
Yeah. So then informing back to public policy of separation, not separation, etc. Do you see ways in which laboratory experiments can give some indication of the potential rate of mutation as a function of how much exposure is between children and the general population?
Yeah, very easily. So one of the oldest tricks in the virology textbook is you can take cells. So in this case, you would take pediatric epithelial cells, and you can actually passage the virus on these cells. So what you can do is infect cells, wait for the progeny virus, take that, infect more cells, wait for the progeny virus, take that, infect more cells. And essentially what you're doing is a selective process whereby you're selecting for viral variants that will just emerge randomly that can preferentially infect the cell type you're using, in this case, pediatric cells. So one thing you could do is you could passage these cells and then passage the virus on these cells. And then say after 10 passages, you could see if that isolate actually replicated much better in pediatric cells compared to the original isolate. And then what you can do is you can actually sequence that virus, and that's very easy and straightforward, see what mutations it has. And then because genomic sequencing has just been so widely used and so powerfully used throughout this pandemic. You can actually go to online databases and say, hey, have any of these mutations that we found in the lab actually emerged in real life SARS-CoV-2 strains? What's their epidemiology associated with that? Were they emerging in children? So you can sort of bridge the gap between the lab and the public policy and the sort of real life situation by doing something like that. So that would be one option.
Okay, thanks. Yeah.
It'd be kind of a cool experiment, actually, now that I think about it. Yanshan, brace yourself for chapter 4 of your thesis.
So yeah, I was thinking about bridging, I mean, obviously we're seeking to make bridges between your lab work and the softer AI, big data work that we're trying to do. So maybe that's a direction.
Yeah, and I think what we're seeing a lot more is those bridges being formed. So some of the other work that we do on SARS-CoV-2, we look at diagnostic markers or prognostic markers, determining whether or not a patient will go on to have severe COVID-19 or mild COVID-19. And one of the major ways that that's done now in the field is machine learning. So, to me, that still remains a mystery, and there's just a section in the paper that's machine learning. But I think you're seeing that divide already being bridged a little bit. And I think we'll see it more and more as more big data sets become available.
Yeah. Thanks.
All right. So thanks very much again, Kirsty, and participants for joining us. And you're looking forward to seeing you in our next seminar. Thank you.
Thank you all.